Can you guys hear me?
Yeah.
Yeah.
Sonia, if you said anything, I did not hear you. Okay. This is gonna be fun. Okay. Yeah, I cannot hear you guys, so this is okay. This is just wonderful. Mm, Okay. I am not sure what's going on with this today. This is okay. Can you guys try saying something now?
Yeah.
Oh my God.
Years.
Okay. That's my idiotic move. Never mind. How can I hear people if the volume's all the way down? Okay. Well, that helps. Sorry about that, guys. Okay. So who wanted to start? Oh, let me allow sharing.
I said I could go first.
Okay. Oh my Lord.
Can you see my screen?
Yes.
Yeah.
Okay, so I had question number 3, which was the cross big G, big E, little g, with little g, little e, and little g produces the following progeny. Big G, little g, 404, little g, little e, 396, little g, little e, 97, and big G, little e, 103. From these data, one can conclude that the big G, and big E loci sort independently, true or false. So first is the background information, and I put that independent assortment occurs during gamete formation, which is segregating pairs of unit factors which assort independently of each other, and all possible gamete combinations should be formed in equal frequency. Linkage is that genes are present on the same chromosome and inherited as a unit and do not segregate at all. Parental combinations would be the same genotype as the parents. Recombinants are a gamete containing a new combination of alleles by crossing over. And then the formula for recombinant frequency is the number of recombinants over the total number times 100. So to use that in the answer, the proportions listed in the question are big G, big E, little e, 404, little e, 396, little g, big E, little e, 97, and big G, little e, 103. And the question is asking if the big G and big E loci assort independently. So the parental combinations are big G, little e, which is 404, and little e, 396. And the recombinants are little g, big E, little e, which is 97, and big G, little e, 103. So to find the number of recombinants, you just add the recombinants together, which is 103 plus 97, so that's 200. And to find the recombinant frequency, you just plug that into the equation, which is the recombinants over the total number. So you do 200 over 1,000 because that's the total number of progeny, and then multiply that by 100, which is 20%. This means that the distance between the big G and the big E loci is 20 map units. Since this is a low recombinant frequency, it means that the low C, big G, and big E are linked. Therefore, the answer is false, because if the genes are linked, then they cannot assort independently.
So I guess my question to you is, what percent of recombination frequency would you need to have one assort independently?
It would have to be over 50.
Good. Okay. Great. Does that make sense to everyone?
Yeah.
Great. Okay. Wonderful. Okay, so I have Kyle and Hind listed for today. Are you guys still presenting today?
Yeah, I can go next.
Okay.
I was working on question number 1, which stated 2 plants, both big A, big B, little b, whose loci are linked in 35 map units apart. What will the proportions of the following gametes from a single plant be? For background information, 1 map unit corresponds to a recombination frequency of 1%. This means if the 2 loci are 35 map units apart, then 35% of the progeny will be recombinant, while the remaining 65% will be non-recombinant. The recombinant gametes are big A, little b, and little b, big A, little a, big B. Since there are 2 different recombinant progeny, we divide the 35% by 2 to get their proportions, which I got 17.5% for big A and little b, and little a, big B. And then I took, and the non-recombinant gametes are big A, big B, and little a, little b. And I did the same thing as above, except divided the 65% by 2 and got the proportions for big A, big B equal to 32.5%, and the proportion of little a, little b equal to 32.5%. And those were just the answers from the previous slide.
Good. Everyone on the same page with this? Guys.
Yeah.
People- oh,
Yeah.
okay.
I can go next.
Okay.
Can you see my screen now?
It's still at started screen sharing at the moment, but cannot see the, oh, now I can, yep.
Okay. So I had question number 2. 2 genes that are 60 map units apart are expected to show independent assortment, 20% recombination, 25, 35, 50, or 60. So the background information, we know that gene mapping determines the order of genes and the relative distance between them in map units. So the recombination frequency equals the distance between the genes. And we already know that 1 map unit equals 1% recombination. So since it's 60 map units, it will equals to 60% recombination, which is answer number F.
Okay, so one thing I need to say is remember when it's above 50%, it automatically goes to independent assortment.
Oh, okay,
Yeah.
I got you.
Does that make sense, everyone?
Yes.
Okay,
Can you
so
explain that?
okay. So with okay. Hin, can you bring back your slide?
Oh, okay.
Sorry. So when it comes to these kind of questions, so when you have, so one map unit is equal to one recombination frequency, right? So it's asking you, so with this question, you would see that with, so if two genes are 60 map units apart, it does mean that it also has 60% recombination, but it's assorting independently because it's above 50%. Does that make sense?
So wait, 50% is the cutoff or something? I don't understand.
Mm-hmm. So anything over 50%, it just says that they wouldn't be linked at that point. So when they're not linked, they end up with independent assortment. Does that make sense?
Yeah, it
Hey,
does.
So if it's 50 map units, would it be 50% or independent?
I think it would be if it's higher than 50%, not equal to, but I need to double check on that again. But if it's anything higher than 50%, it would be independent assortment. Does that make sense?
Yeah.
So since we're kind of ahead of schedule, instead of me going through the next topic and make you guys more confused, I planned a little game for you guys. Let me just get this work. Okay. Okay, let me share this. Can you guys see my screen?
Yes.
Okay. So we're gonna play a game of Kahoot to review for the exam. So once you guys are all in, this is also counting as your attendance. So
hey, so wait, we have to join that with the app?
yeah, with the app, or you can go online here.
All right.
Missing like 2 more people here. Who's missing? Okay. Let me just see. I think I'm still missing one more person. Who am I missing? Mm, 22 people. Who's missing? Oh, perfect. Great. Okay, let's start. So what term is used to express the idea that several genes exert influence over the same characteristic? Okay. So yes, gene interaction is correct. I don't even know where I pulled coextension from. So this is the definition, guys. So makes sense, right?
Yes.
Okay. Ooh, okay. Cool. In codominance, both phenotypes are expressed in heterozygotes, true or false? Okay, so codominance, yes, both are expressed. Both phenotypes are expressed in heterozygotes. Just make sure you guys remember that part. There's the difference between incomplete dominance and codominance. Incomplete dominance is where you have the intermediate, right? And then codominance, it's like the blood groups, AB, like the AB blood type, right? We're cool with this one, right?
Yeah.
Yeah,
Okay. In incomplete dominance, the heterozygote shows an intermediate phenotype between two homozygotes. Okay. Okay, so like we just talked about, incomplete dominance, you see the intermediate. Okay. Okay. Let's go to the next question. With incomplete dominance, a likely ratio resulting from a monohybrid cross would be. So, what do you guys think? Oh, okay. We have a little bit of a majority wins, yes. It's a 1-to-1-to-1 ratio. So when we're talking about incomplete dominance, guys, never choose these two because these two involve two genes, right? When we're talking about dominance, we're only looking at one single gene. So that would narrow it down to these two, right? And then we know that this is a regular monohybrid phenotypic, right? And then this guy is where it comes in with incomplete dominance because you have two heterozygotes that look completely different from the homozygotes. Does that make sense?
Yes.
So if a question comes with incomplete dominance, make sure you guys remember that, okay? It's the intermediate, so it becomes a different phenotype, right?
Yep.
Okay. When a cross is made, complete linkage is shown when offspring show parental and recombinant genotypes. I have a lot of people saying true. So guys, complete linkage means that there is no recombination occurring between them, right? Like there couldn't be any, like, or it would be very low to see any. So you would only see the parent genes, right? Does that make sense?
Mm-hmm. Can you say that again?
So with when it says complete linkage is shown, you only see the parental groups, right? So not, you don't see recombinants at that point. Does that make sense?
Yeah.
Because they're so close together that there's not enough room to actually cross over and make recombinants, right? You would end up with half of them would end up one of the genotype, one of the parent genotypes, the other will be the other parent genotype. No recombinants. Everyone cool with this?
Yep.
The phenomenon in which a single gene influences the development of many phenotypes is called what? Okay, so that is pleiotropy. When one single gene can affect different traits. So remember that slide where I had a trait going on and it's like, hey, this gene, that gene, other gene, and all that. So that's the definition of pleiotropy, right? Epistasis is controlling it. So one gene is controlling the other gene, right? Does that make sense?
No, wait, so epistasis, one gene can't control several genes?
So this question is asking, what is the term that refers to a single gene affecting different phenotypes, right? So that's pleiotropy. Epistasis is when one gene controls another gene itself, not the phenotype per se. Does that make sense?
Alright, so just the genotype.
Yep.
But doesn't that affect the phenotype too?
Yes, but in this case, so epistasis is just it does affect the phenotype as well. However, it's one gene masking one other gene, not giving rise to many different phenotypes. We're just sticking to one phenotype in the case of epistasis. Whereas pleiotropy, you end up with one gene controlling a whole bunch of phenotypes. So it's like, for example, one little gene can be controlling hair color, eye color, skin color, depending on whatever trait it's going on. So that's kind of what they mean by pleiotropy. Does that make sense?
Okay.
Okay, good. True or false? When a cross is made, independent assortment is shown when the gene, when the offspring show only parental genotypes. Okay, so false is definitely correct. So guys, so it's a similar thing with independent assortment. They're far enough that you can actually see some recombination going on. So you will actually see parental groups and recombinants. Does that make sense?
Yep.
Great, okay. Two genes that are separated by 20 map units show a recombination percentage of 20%. Oh. It is true. What happened here? Okay. So as we know, one map unit equals to 1% recombination frequency. So that ends up being 20 map units would be a recombination percentage of 20%. I think we're cool with that, with majority. Okay. Oh, you're on a streak, Megan. Good. A condition in which one gene pair masks another, the expression of a non-allelic gene pair is called no? Okay, so epistasis is correct. Any of the dominance ones, again, guys, when you're talking about dominance, codominance, incomplete dominance, you're only talking about one gene pair, right? So you're talking about the alleles within that gene pair. When you're talking epistasis, that's when one gene pair is controlling another gene pair, okay? You cool?
Mm-hmm.
Good, okay. The mapping of an individual is called good. DNA fingerprinting, that is more identifying people using DNA. Recombinant DNA is just DNA that's recombined, and you can use that for different techniques. Karyotyping is what you use to map each individual chromosome, okay? Traits controlled by genes on the X chromosomes are blank traits. Good. That is sex linked. So X chromosome, guys, it's always sex linked. Polygenic just means multiple genes. That has nothing to do with the X chromosome. Autosomal is more other traits that are not on the X chromosome. So anything that's not on the sex chromosomes are autosomal chromosomes or autosomal traits. Okay? Does that make sense?
Yes.
Okay, good. Okay, let's. What is the useful purpose of chromosome maps? Oh, okay, we have a pretty much an even spread here. So, but majority of you guys answered correctly. It is mapping genetic diseases. It is, so chromosome maps. Okay, so I guess when they say chromosome maps, I think in this question it meant like karyograms or karyotyping. So when you look at a karyogram itself, you can't directly visualize the distance between these genes on a chromosome or the distance itself, actually. But you can tell different genetic diseases because you can tell if there's some kind of abnormality going on, which that's kind of in the next module at that point. So good. Whoa. Okay. Lena the nematode is disappointed. So Protonor, guys. Oh, wait, something in the chat. Yes, Lena the nematode is very upset with you guys now. So it is XXXO. Don't forget it. Thankfully, Lena's hiding right now. So you guys are on the good side right now. But don't forget it, please. If we have Lena upset, it's not a good day for anyone in this lab. So So remember, guys, this is the other one. What is the other one called? What is the other mode called?
I think it's called normal.
That's not the actual name of it, though. Or we can just go to the next question. Yay. So yes, the XXXY scheme is the ligase mode, okay?
Yep.
Good. If this comes up and everyone gets it wrong, I will be sad. Lena will be sad too. Chromosomes that are not involved in sex determination are what? Good. Okay, so homologous and analogous, those are just saying like the relationship between two different chromosomes. Sex chromosomes are the ones with sex determination, and then autosomes are not, obviously not doing that. I think we have time for like one more question. Sex determined varies between animals. It's like the easy question. Yes, that is true. It varies. So it depends on the species or animal, which sex chromosomes you have and whatever else is there. Does that make sense?
Yes.
In humans, sex determination is generally depending on good. It all depends on if we have a Y chromosome or not, right? Because you can have many, many X's, but if you don't have a Y, you will never be male. All of the following genes are involved in male development except oh. Okay. So we know these two, this one is definitely there. Even so, without even knowing any of these guys' without knowing these two, you can automatically think of this guy because X is part of the X inactivation center. So that is not in males, okay? That is only in females with their X chromosomes, multiple X chromosomes, right? Does that make sense?
Yeah.
So I think we'll have to stop there because we're out of time now. Okay, so let me just. So basically, FGF9 is an autocrine and paracrine prostatic growth factor. So it's essentially helping the prostate develop, okay? Any other questions?
What is a XIST?
So XIST is one of the genes that is involved in, what's the word? X inactivation. So remember how we were talking about X inactivation the previous week?
Yeah.
So it's one of the main genes that are involved in that process, in the mechanism itself.
Okay.
Any other questions, concerns? Do you guys like Kahoot?
That was fun.
Yeah, that was fun.
Yes, I
Yeah.
love Kahoot.
I'm glad. This was my first time ever trying Kahoot for a genetics session, so
I feel like it's a good way to review like before exams and stuff.
yeah, it's definitely gotten everyone engaged. So
I got really excited as soon as I heard the theme music.
I'm glad. I'm glad you guys liked it.
Is there
But
a
if you
quiz?
guys have any questions, let me know. Your exam's coming up next week before we meet again, so
Is there a quiz due this week?
there is a quiz due tonight, quiz number 5. There will be one up for that's due next week, but I might extend it a little bit just because you know, exam and whatnot. So maybe make that one due like next Friday or so. Okay?
Okay,
Great.
Okay, thank you.
Yep.
Thank you.
thank you.
Yep.